Thank you, Dr. Ansela, for the introduction, and welcome everyone from Heidelberg. My part is to give you an introduction into some of the basic principles behind targeting BCMA in multiple myeloma and introduce you to the seminal clinical trial data that led to approval of these BCMA targeting agents. So BCMA has really emerged as the major immunotherapy target in multiple myeloma over the recent few years. And this is linked to its crucial role in multiple myeloma biology and its largely specific expression in plasma cells. Upon ligand binding, BCMA activates the NF-kappa B pathway, which is associated with tumor cell proliferation, drug resistance, and promoting an immunosuppressive tumor microenvironment. BCMA is generally expressed stably across disease stages and lines of therapy in BCMA-naive patients, I have to say, and it is minimally expressed outside of plasma cells and maybe late-stage B cells. BCMA also has a value as a biomarker in that it is being shed from the plasma cell membrane and occurs as soluble BCMA in the plasma, where it can be measured and is associated with both prognosis and also disease response under therapy. Targeting BCMA is a major strategy in immunotherapy now, but it may ultimately be limited by loss of targetable expression of BCMA, mostly due to either genomic deletions of the BCMA locus or point mutations and some other mechanisms that are less well characterized at this point. So these are the modalities that we have currently available to treat our patients targeting BCMA. The first one being antibody-drug conjugate, basically an anti-BCMA monoclonal antibody that is conjugated to a cytostatic payload, and it delivers this payload directly to the myeloma cell, thereby sparing excessive toxicity. Second modality are bispecific antibodies shown in the bottom of the slide, which are antibodies with two targeting moieties, one targeting BCMA on the myeloma cell, and the other one usually targeting CD3 on T cells, kind of functioning as an adapter between T cells and myeloma cells, recruiting T cells towards myeloma cells and triggering their cytotoxic activity. And then third of them are CAR T cells, which are genetically modified autologous T cells that express an artificial receptor that can bind to BCMA and thus leads to binding of cytotoxic T cells to myeloma cells and triggers their cytotoxicity directly. This is an overview of the currently approved therapeutic options targeting BCMA. On the right side, you can see the exact indications according to geographic location, with major difference, or with minor differences, I should say, between the US and Europe. And I will not go too much into detail with this due to time limitations, but I believe that all of you will have the slides after the symposium, so you can look into this in more detail if you wish. So in the ADC space, we have belantamab mafodotin, or belamaf, which is the only of these BCMA targeting therapies that is approved in combination with other agents, VD or Pomdex, and it's approved in the early relapse space in first or in second or third line. Then in the bispecific antibody space, there are three choices: teclistamab or TEC, elranatamab or ELRA, and linvoseltamab or LINVO. All of these are improved in later lines of therapy, typically after 3 or 4 prior lines of therapy, and they all have very similar efficacy and toxicity data, I should say. Maybe the main practical difference between those agents is their mode of application, with TEC and ELRA being subcutaneous injections, while linvoseltamab needs to be infused. And then third, on the CAR T cell space, we have 2 products, one of them being idacaptogene viclucel or ide-cel, and ciltacabtagene autoleucel or cilta-cel, and both of them are approved for treatment of early relapse in either after 1 prior line of therapy or 2 prior lines of therapy. So having set the stage, I want to jump right into the top-line clinical trial data from these agents. Here we're starting with belamaf, and there are 2 recent phase 3 trials comparing belamaf against other common regimens in multiple myeloma. This one is the DREAM-M7 trial. And as you can see, the overall response rates in this early relapse patient population are high, or above 80% for the belamaf combination. And this results in a marked PFS difference in favor of the belamaf combination of roughly 37 months against 13 months for the daratumumab combination. So this led to a hazard ratio of 0.41 for this combination and to approval basically in second line. As you can see, besides the common toxicities in the hematologic AE and infection AE space, which are fairly similar between arms if you consider the different treatment durations, the major AE that is associated with belamaf are really ocular toxicities. And these basically lead to blurred vision, dry eyes, cataract. This can be fairly burdensome for patients, but it is considered manageable through dose modifications, basically by reducing doses or holding the next infusion and waiting for it to disappear. These AEs are virtually all reversible. This is the second of the belamaf trials, DREAM-M8, and we see fairly similar results, overall response rates of the belamaf combination, again, close to 80%, resulting now in a 12-month PFS difference of roughly 70 versus 50% in favor of the belamaf combination. Again, the hazard ratio is close to 0.5, and in terms of adverse events, we see the same pattern, basically blurred vision, ocular toxicity occurring as a specific feature associated with belamaf. Again, this was manageable with dose modifications, and importantly, efficacy is maintained despite these dose modifications. So now jumping into the bispecifics arena, these are the MajesTEC-1 study results for teclistamab. This was a phase 1/2 study, so no control arm, but I think the results speak for themselves, considering this is a late relapse trial in a heavily pretreated patient population with a number of prior lines of therapy of 5. You can see high response rate in the 60% range, a median PFS of close to 1 year, and very low rates of higher grade CRS or neurotoxicity. The major toxicity associated with this treatment being infections. Those can be higher grade, and they necessitate post-monitoring and prophylactic measures such as antibiotics and immunoglobulin substitution. So very similar data for elranatamab, the MagnetisMM-3 trial. Again, high overall response rate translating into durable responses, 15-month PFS here, close to 50%. And again, low rates of CRS and ICANS, but fairly high rates of infections in the grade 3 to 4 space. And again, similar data for linvoseltamab, the linker MM1 phase 1/2 trial. Again, higher overall response rate, median PFS not reached so far, but at the 12-month PFS point, we have a PFS of 70% currently, and patients responding can keep responding for much longer, as you can see by the median duration of response of close to 30 months. Again, the toxicities are similar between these bispecifics, with infections being the major clinical concern. And with this, we are moving to the last type of drugs, the CAR T cells. This is the data for ide-cel, both the KarMMa trial in the heavily pretreated space, and then the larger phase 3 KarMMa-3 trial in the less heavily pretreated patients. As you can see, overall response is around 70% in both collectives. Median PFS is roughly around a year, a bit longer for the less heavily pretreated patients. And we again see fairly low levels of CRS and neurotoxicity, at least at the grade 3 to 4 level, 5 and 3% in the more heavily pretreated patients, and 1 and 3%. So a little bit lower in the less heavily pretreated patients, which is probably related to better disease control before the CAR T infusion. And so the last one, cilta-cel, again, we have the heavily pretreated cohort in the CARTITUDE-1 trial on the left side, less heavily pretreated early relapse patients in the CARTITUDE-4 trial on the right side. And now we see even better overall response rates, I would say, around 90%. And this translated to a 12-month PFS in the CARTITUDE-1 trial of 77%, and 12-month PFS in the CARTITUDE-4 of, again, 76%, close together. But with additional follow-up, we start seeing, and it's not on the slide, but at the 30-month point, we see basically 60% PFS with CAR-T2 in the less heavily pretreated patient in the earlier lines, which is better than what we see in the late lines. And still in the late line setting, after 5 years, we still have ongoing PFS MRD negativity in a third of the patients. And this is very remarkable in this heavily pretreated patient population, basically fueling speculation of cure in these patients. And lastly, I want to mention the CRS and neurotoxicity rates, which seem to be a bit higher here than with ide-cel. So the higher efficacy is also bought by somewhat higher toxicity. But again, in the earlier lines of therapy in the CAR-T2-4 trial, this rate of CRS and neurotoxicities was a bit lower again. And with this, I'm coming to the take-home messages. BCMA-directed therapies have expanded the treatment landscape for relapsed refractory multiple myeloma, providing effective options for patients regardless of risk status. Each therapeutic class offers distinct advantages and challenges. Current ADC combinations provide an accessible option but require management of ocular toxicity. Bispecific antibodies offer off-the-shelf availability and flexible dosing, but require close monitoring of treatment-related toxicities, particularly infections, which often necessitate prophylaxis and immunoglobulin support. CAR T-cell therapies can induce deep and durable remissions following a single infusion, but require close monitoring, specialized centers, and extended manufacturing time. Bispecific antibodies in CAR T-cell therapy rely on T-cell-mediated cytotoxicity, whereas ADCs deliver a chemotherapeutic payload. The distinct mechanisms of action and resistance enable sequential use of BCMA-directed therapies. However, all can eventually be limited by antigen loss, mutation, or downregulation. Management of treatment-related toxicities remains critical to optimizing patient outcomes across all BCMA-directed modalities.